Welcome to Cloud-ATAC, an open-source, cloud-based, interactive pipeline tutorial for differential ATAC-Seq footprint analysis. This module will demonstrate an example analysis pipeline for bulk and single-cell ATAC-Seq data implemented in Jupyter Notebooks on the cloud. The purpose of these tutorials is to help users familiarize themselves with the analysis steps involved in processing ATAC-Seq data, including special considerations for single-end versus paired-end sequencing, as well as for single-cell sequencing. These tutorials cover specific step-by-step workflows, starting with downloading raw sequencing data and extending to differential peak identification, genome annotation, and footprint analysis. This module involves interactive quizzes and customized visualizations. This module is composed of 4 lessons using examples from publicly available data. To reduce the time requirements, we provide specially prepared downsample data. We encourage going through these modules with the example datasets first, but you can also use these notebooks to process your own dataset. The first lesson will introduce the dataset and proceed through quality control, trimming, alignment to the genome, and deduplication. The second lesson will include visualization of ATAC-Seq signal, profiling of insert sizes, and peak identification. The third lesson will include differential peak identification, peak annotation, and footprint analysis. Now, the fourth lesson focuses on single-cell ATAC-Seq data and includes steps similar to bulk analysis, as well as special considerations for single-cell. During these lessons, we will use a publicly available ATAC-Seq dataset to evaluate differential accessibility footprints that occur after depletion of the BAF chromatin remodeling complex. The BAF complex is essential for differentiation, and understanding the impact of BAF on chromatin accessibility has been key to identify transcription factor binding sites that play a role during differentiation. This is just one example of how ATAC-Seq can be used to further our understanding of important biological phenomena. Another study performed ATAC-Seq in 410 different cancer samples belonging to the Cancer Genome Atlas and identified distinct regulatory landscapes among different cancer types. Using this data, they were able to find differential footprints that define transcription factor activities in cancer subtypes. In another example, ATAC-Seq has been used to define chromatin features that are inherited and persist from gamete to embryo and revealed greater accessibility in sperm than previously expected. These studies, along with numerous others, provide a wealth of information that can be used to define new hypotheses, support current research, and drive innovation forward. Therefore, learning to analyze these datasets effectively is an important skill set useful in a wide range of research areas. ATAC-Seq investigates the accessibility of chromatin using TN5 transposase. During the ATAC-Seq experiment, TN5 inserts preloaded DNA sequencing adapters. This insertion occurs in accessible chromatin and is blocked by protein-occupied DNA, such as nucleosomes or transcription factor binding sites. At these loci, insertion occurs next to the occupied site, which results in fragments of various sizes. These sizes range from small fragments protected by transcription factors to larger fragments consistent with the size of mononucleosomes, dinucleosomes, and so on. ATAC-Seq data can be analyzed to measure accessibility in general, but can also be used to delineate between transposase hypersensitive sites that are bound by transcription factors versus nucleosome-occupied loci. Because TN5 insertion occurs proximal to protein binding, it can also be used to generate an accessibility footprint at specific transcription factor binding motifs. The lessons in this module will introduce basic concepts and considerations for ATAC-Seq analysis. We recommend starting with Tutorial 1, Preprocessing Quality Control, but each lesson can be taken independently. The steps are implemented in Jupyter Notebooks organized into blocks of code and marked down comments. To run the code, click on a section and select the Run button at the top. These lessons also contain quizzes, flashcards, and HTML reports that can be interacted with by clicking on them. At the end of the lessons, you will be able to run a basic ATAC-Seq analysis pipeline, understand special considerations for single-end versus paired-end sequencing, and single-cell versus bulk ATAC-Seq analysis, perform differential analysis to identify changes in accessibility, and visualize results in several ways, including how to plot signal across the locus of interest, create average profiles at genomic loci, including promoters, perform motif and footprint analysis, and get clustering of single cells based on accessibility profiles. This module will introduce a pipeline for a bulk ATAC-Seq analysis as well as for single-cell ATAC-Seq. Each has its own advantages and uses, and the user should decide which will be most fitting for their research question. While bulk ATAC-Seq examines the average accessibility within the sample, single cell can identify different populations of cells with distinct accessibility profiles. This makes single cell ideal for epigenetic studies in rare cell types or in samples with mixed cellular populations. Bulk ATAC-Seq analysis can be performed with basic molecular biology tools and kits, whereas sample preparation is more complex and sometimes requires special equipment for single cell. Lastly, bulk ATAC-Seq analysis is computationally simpler and relatively fast, whereas single cell can be more complex and take a few days of processing time when using CPUs. However, recent innovations have shortened the processing time and through the use of GPU-based computing. In this module, we demonstrate the use of GPUs for single-cell ATAC-Seq analysis. In the next part of the video, we will cover more information concerning single-cell ATAC-Seq.
Welcome to Cloud Single Cell ATAC-Seq, an open-source interactive pipeline tutorial for differential ATAC-Seq analysis. In this video, we illustrate the usefulness of conducting single-cell ATAC-Seq analysis in a Google Cloud environment by utilizing Jupyter Notebook and the Rapids pipeline. I will also delve into the importance of performing ATAC-Seq and highlight the significance of utilizing this technique on single-cell samples. I will walk through the key steps involved in sample processing, including the dataset and pipeline that will be used in this demonstration. Additionally, I will provide a detailed explanation of the various steps involved in conducting a single-cell ATAC-Seq analysis. We will now examine an example dataset and the pipeline that will be utilized to analyze single-cell ATAC-Seq data. We will utilize the droplet single-cell ATAC-Seq dataset comprising 60,000 cells obtained from human bone marrow samples for our analysis, comparing between the control and stimulated conditions. We will utilize the Rapids pipeline to conduct downstream analysis of single-cell ATAC-Seq data. Rapids pipeline is a set of open-source libraries and frameworks that allow for fast and efficient data processing and analysis on GPUs. It is designed to accelerate data science and machine learning workloads by leveraging the power of NVIDIA CUDA-enabled GPUs. Utilizing GPU technology in the Rapids pipeline drastically reduces processing time, cutting it down from days to just hours. The Rapids pipeline includes libraries for data manipulation, data visualization, machine learning, and deep learning. It also includes tools for data loading and to perform extract and transform operations, allowing you to easily move data between different stages of the workflow. In this section, we will delve into the detailed procedures and techniques used in the downstream analysis of single-cell ATAC-Seq data. The ATAC-Seq analysis process for single-cell data includes several important steps, including quality control to ensure data integrity, normalization to adjust for technical variations, dimensionality reduction to identify key features, clustering to group similar cells, annotation to assign functional information, peak calling to identify regions of active transcription, identification of variable peaks to identify cell-specific patterns, motif analysis to uncover transcription factor binding sites, prediction of interactions between regulatory elements, and pseudotime analysis to track developmental progression. And before you embark on performing some of these steps, it is important to understand the Google Cloud framework. You will be utilizing the Rapids pipeline to analyze single-cell ATAC-Seq data, which is stored on a Google bucket. The pipeline will pull the datasets from the bucket and generate annotation data, enabling multimodal and multi-omic analysis of the single-cell omics data. This will allow for efficient storage, querying, and analysis of various data points within the dataset. Let us look at the implementation of RAPIDS pipeline in Jupyter Notebook. In the first step, you will set up a Conda environment and install the necessary packages, including RAPIDS AI and other GPU packages to prepare for importing and working with the dataset. In step 2, you will carry out essential pre-processing and quality control measures to ensure the integrity and accuracy of your data. This includes setting appropriate peak calling parameters and utilizing techniques such as principal component analysis to reduce dimensionality and enhance data interpretation. In the third step of the process, you will conduct downstream analysis by utilizing a combination of clustering and visualization techniques such as UMAP and t-SNE to identify patterns and trends in the data. Additionally, you will identify peaks that are located near overlapping marker genes and perform a differential peak analysis to determine any significant differences between the peaks. In conclusion, single-cell ATAC-Seq is a powerful tool for understanding the complexity of cellular systems and in unraveling the underlying regulatory mechanisms of gene expression. With its ability to provide detailed information on the chromatin landscape of individual cells, this technique has the potential to revolutionize our understanding of disease and aid in the development of more targeted therapies. We are excited to see the continued advancements in this field and the impact it will have on the future of biomedical research.